Hello all, my name is Jason Wong, and during this REU, I worked with Dr. Panagiotou in the Department of Mathematics at the University of Tennessee, Chattanooga. The overarching goal of our project was to use topology to characterize protein folding rates. Specifically, we use methods derived from topology to measure the complexity of a protein's 3D shape and compare the folding rate to those measures. Let's begin with a short introduction to topology and the concept of knots. So if you take a string like this and twist it around itself a little bit, maybe loop it through itself, basically do whatever you want except cut it and glue it, and finally join the ends, then you have formed a knot, kind of like tying an actual knot. A goal of knot theory is to determine which knots are identical. On the slide, we have two of the simplest knots. The left is a trefoil and the right is a figure 8. These are distinct knots, meaning you cannot transform one into the other without cutting or gluing them. I can twist this string around itself all I want to form a seemingly complex knot, for example, but this knot, when I untwist it, is going to be equivalent to just a circle. Now, open knots are closed knots but with open ends. Because any open knot can be undone into a straight line, they are all topologically equivalent. However, open knots are still distinguishable by how tangled they are. These two open knots on the slide are formed by cutting out a piece of the trefoil and figure 8 knots, respectively. They still resemble their original structures and are easily distinguishable. For example, the left knot has 3 points of intersection, or crossings, and the right knot has 4. We will now discuss how proteins can be modeled as open knots. So proteins are chains of amino acids, and each amino acid has a central carbon atom known as the alpha carbon. We can represent any protein as an open knot by replacing each amino acid with a point at the alpha carbon and connecting all of those points together. We use coordinates from the protein data bank in order to define this structure. Then we can perform calculations on that structure to measure its complexity. This figure on the right shows the protein 1v9e as its image from the PDB. The structure is determined by the amino acids that compose the chain. To perform their biological function, each protein must take on a very specific three-dimensional conformation called the native state. To reach the native state, proteins go through a process called folding. The figure on the right is a model of 1v9e in its native state. Now, K sub f is the experimentally determined folding rate constant. For our purposes, we compare our measures against the natural log of K sub f. Folding can occur in a two-state mechanism, meaning that protein is either fully folded or unfolded, or in a multi-state mechanism, meaning the protein can get stuck in transition states while folding, and this folding style influences the folding rate. For our study, we used folding rate data for a set of two-state and multi-state proteins used in a previous study by Broom, and we used coordinate data from the PDB. Now, folding is a very complex process, and the problem of how proteins fold is very broad. This project focused on understanding the properties of the native state, specifically how its complexity is related to the folding rate. We want to find a reliable way to predict the folding rate given the 3D structure of the native state. It is hypothesized that the folding rate decreases with increasing complexity of the native state. The measure we use to quantify the complexity of the folded state is the second Vassiliev measure, or V2. To calculate the V2 of this trefoil knot, first you must identify all of the crossings, so 1, 2, 3 here. And then for every pair of crossings, you check for the following pattern. So you go over, and then under, and then back under the first one, and then back over the second. And once you have checked that, then you can find the sign of the crossing for each pair. So both of these are -1, and then you take their product to be +1. And you do this for every pair of crossings on this knot. So for this trefoil, there is another pair with a product also of +1. And finally, you can calculate the V2 by taking 1/2 of the sum of all of these products. So here, V2 equals 1. Now, V2 remains the same no matter how the knot is represented. For example, the figure on the right is topologically equivalent to the trefoil, and so V2 equals 1 for that knot. For open knots like proteins, V2 is defined as an average over all possible projections. Each specific way the open knot is viewed has an associated V2 value, and the actual value is an average of all of these. There are infinitely many projections, so we approximate the value by randomly selecting projections. The figure on the left is one projection of 1v9e, where V2 equals 0, and the figure on the right is another projection where V2 equals 1. So from these two projections, we could estimate that V2 equals 0.5. Now, to get a precise estimate, we sampled 5,000 projections for multistate and 10,000 projections for two-state proteins in the dataset. With 5,000 projections, the estimated V2 for 1v9e is about 0.8. Now, the absolute Vassiliev measure, or AV2, is very similar to V2, except we average the absolute value of V2 for each projection. And in addition to these measures, we also calculated V2 over N and AV2 over N by dividing the measure for each protein by its length. It is known that V2 can depend on the length of open chains, so dividing by the length might provide a more accurate measure. V2 is a continuous function that depends only on the coordinates of the chain. So V2 and AV2 measure the resemblance of the protein to a knot. All of the native states of the proteins we used have V2 measures between 0 and 1, which we can imagine as measuring how much of a knot the protein forms on a continuous scale from the unknot to the trefoil knot, keeping in mind that proteins are open and not closed. Now, before we see the results, I want to quickly summarize our methods. Previous studies have determined the folding rate and the structure of the native state of a set of proteins. So we model the folded protein as an open knot, and then approximate V2, which is a measure of complexity of the shape for all proteins in that dataset. And finally, we plot the folding rate against V2. So this slide compares the folding rate to V2 and V2 over N. The correlation coefficients are both about -0.36. The blue points represent the two-state proteins, and the orange points represent multi-state proteins. The majority of proteins have a V2 of around 0, forming this large block of points right here, where there is little knotting character present in the folded structure. Interestingly, the V2 values are all positive. However, there do exist specific projections whose V2 value is negative, and those would cancel out some of the positive values when averaging, which is undesirable, motivating the use of AV2, whose results will be shown on the next slide. So this slide shows the results correlating the protein folding rate with AV2 and AV2 over N of the folded state. The correlations here are about -0.4 and -0.43. These plots are very similar to those on the last slide, but comparing the correlation coefficients between the plots shows us that AV2 correlates better with the folding rate than V2 does. Now, this heat map summarizes all of the correlation values. For example, the correlation between the protein folding rate and AV2 for two-state proteins would be -0.61. This map shows us that AV2 and V2 are moderately good predictors of folding rate, with AV2 being slightly better. We also see that correlations for each individual set are better than the combined set, implying that the folding kinetics are important to predicting the folding rate. Protein folding can be thought of as a search for the native state. there is a large ensemble of shapes a protein can go through before reaching the native state. The correlations we saw earlier motivate the following hypothesis. More complex structures have a lower probability of occurring, and the folding rate correlates with the probability that the protein will reach a conformation of complexity while folding equal to the complexity of its native state. To test this hypothesis, we try to predict folding rate using the probability of attaining a specific value of V2 during folding. The approach is to create a probability density function, or PDF, of V2, and then compare the folding rate to the PDF value of V2 of the native state. We obtained data from a research group at the University of Chicago, led by Dr. Sosnick, that simulated the folding of 50 proteins. For each protein, they obtained the coordinates of 2,750 random conformations during folding. We estimated the V2 of each of these conformations using 1,000 projections and plotted their frequencies on a histogram. Then we fit the histogram with a distribution, the result of which will be shown in the next slides. The function is then normalized to produce the final PDF, which indicates the probability of attaining any V2 value during folding. Then for each protein in the Broom dataset, we plotted folding rate against the probability density function of the V2 of the native state, and did the same with the other measures. So this graph shows, using a logarithmic scale, the number of conformations in the simulation data that had each specific V2 value. There are values outside this range, but those occur very sparsely. The histograms were fitted with this black curve, and then the probability density function is created by scaling that curve to make the area underneath equal to 1. Now this plot was generated using the same method, except using AV2 as a measure. The shape is very similar to the positive side of the previous plot. Interestingly, the distributions for individual proteins are very similar in shape to this overall distribution, which could indicate that similar proteins go through topologically similar conformations while folding. The same technique was used to fit the histograms for V2 over N and AV2 over N data. Almost all values lie in this small region here. Keep in mind that this is plotted on a log scale, so although these bars over here look tall, they only represent about 10 values at most, compared to 100,000 values here. Since the distribution for V2 is similar regardless of the protein, this distribution could be influenced by the length of the proteins being used. Now we will see the final results. The following plots show the folding rate as a function of the probability density of the V2 of the native state. Now this plot compares the folding rate to the PDF of AV2. Note that although probability values must lie between 0 and 1, the PDF has no such restriction. A PDF value of about 300 at AV2 equals 0, for example, means that the probability of a random conformation having an AV2 between, say, 0 and 1,000th is about 300 times 1,000th, or 0.3. We see a slightly stronger correlation plotting the rate against the PDF than when comparing the rate directly to AV2, about 0.5 here versus negative 0.4 earlier, meaning that the PDF is a viable predictor of folding rate. The inset plot depicts the same data, but for PDF values between 0 and 30. This low probability region corresponds to proteins whose native state has a high AV2. And finally, here we have the folding rate plotted against the PDF values for AV2 over N. This overall correlation is worse than AV2, about 0.37 versus 0.5, but the zoomed in region shown in the inset plot has a better correlation than the overall plot. so AV2 over N might be a better predictor in that region. In the direct comparisons earlier, AV2 over N had a better correlation with the folding rate than AV2 for this combined set. At this point, we are not sure what significance that dividing the measure by the length has. This map here shows all of the correlations that were calculated from this second approach. 0.21 here, for example, means that for the set of multi-state proteins, the folding rate showed about a 0.21 correlation with the PDF of AV2 of the native state. The correlation with multi-state proteins here is quite poor. The distributions work much better at predicting two-state folding rates. And conclusion? So the big takeaway from our study is that all of the measures were able to correlate with protein folding rates. The overall set gave R values of up to 0.4 with the direct comparison and up to 0.5 with the probability distribution comparison. We saw that dividing by the length could result in better or worse measures, so we would like to determine their significance in the future. Though the measures we used have good correlation with the folding rate, they take a lot of time and power to compute, especially to create the PDFs. So other measures might be more practical to use, but still our results provide further evidence that proteins with complex native states do take longer to fold and provide another tool to predict the folding rate. The next step in this project would be to actually apply these measures to predict folding rates, which would allow us to compare them to other measures. We have done one case so far with the proteins 2JWU and 2JWS. They are the same protein, but they differ at 7 amino acids, which causes one to fold 100 times faster than the other. We wanted to see if our measures could predict this large difference. The fit created using our first approach, direct comparison, was unable to predict any difference. The second approach, comparing to the probability density function, predicted a significant difference that the topologically simpler one folds about 10 times faster. This is not a bad result, but it is still far from an accurate model. We would like to test in the future all 4 metrics with other cases and see what results we get, so we can compare them with other topological measures that predict folding rate. So I had an excellent experience performing this research over the past 10 weeks, and I would like to thank Dr. Panagiotou for being a great mentor throughout this project. I would also like to thank the NSF and UTC for funding my project, Dr. Chen for giving me the opportunity to do this research, and my family for supporting me throughout the summer. These are some of the references that I used during this research project. Thanks to all of you for coming to my presentation.